CVHS. It's good to see everyone here. Good to see old faces and new faces, and I'm really proud to be here and starting off kind of the resurgence of CVHS. I am Ben Witzke. My background is as a scientific illustrator. I've always loved herpetology and herps. I got involved with this group years ago through kind of their field herping gang, I guess. There was a pretty tight group of folks. And then through that, sort of met folks who were professional herpetologists through Mike Sutton, who some of you may remember. He had a huge collection of native herps. And when I started finding stuff that didn't belong, at least I didn't know what it was, and started looking into it, he helped connect me with institutions like Academy of Sciences and the MVZ at UC Berkeley, which has sort of helped segue me into a job as a professional biologist. My degree is in art, not in biology. I did study biology a little bit in university, but I was a terrible student. So I got a degree in illustration and then a master's in science illustration with the idea of doing science communication, and yeah, sort of segued that into a job taking care of frogs for the zoo. As I describe it, farming frogs for the government. So my sort of personal passion in field herping is plethodonid lungless salamanders native to California. And I like the most boring salamanders that are out there, the slender salamanders. I find them fascinating, not because they're bright or shiny, but just because when you actually start to study their native ranges and groupings of sort of biodiversity within that genus, you start to kind of, it's like you can look back through time because of the way that our earthquake fault lines and glaciation in the last ice age sort of pushed things around and then left them there. So that's kind of what fascinates me about them. Not that I think they're super spectacular to look at, as this is some sort of Batrachoseps related illustration work of mine. And that's actually a Hydromanes, that's a web-toed salamander, which are also some of my favorites. And that one is possibly a new species. They're still working on it.
COVID messed that up.
Huh?
I'm still sitting on shit tons of photos of them.
Yeah, I still haven't, I've been down in those caves and I still haven't even seen one in person.
Awesome.
And I'll come back and talk more about cool salamanders all over the place, but yeah, there was a bunch of new slender salamanders discovered in the desert this last year, and a brand new Hydromandes, which is not even split out like the Shasta ones from known species through genetics. It was like, we're gonna go to this mountain, it's, you know, tens of miles away from any known population. We think it's here it is, and it looks completely different. It's very cool. Not my work, but I was very happy to know about it. How'd that get in here? So sort of as a hobby, no one ever pays me to do this. I go around the state and I look in weird places for salamanders that may or may not be there. I was part of a team several years ago that went to look for the relictual slender salamander. There was one known site forever, and then Academy of Sciences discovered a second. We went out and discovered a third, and since then they've discovered 7 or 8 more. That's the one up on the left there. The thing that's really cool about those slender salamanders is they actually breed aquatically, and you can sort of see it in that photo. They have a slightly flattened tail that kind of aids them in that, but they lay eggs right in the water column, and they're the only slender salamander to do that. And then all the rest of these are fairly rare slender salamanders. And that one on the right is actually the one I found out in Knight's Ferry that sort of started this all off for me and set a fire. Because I didn't know what I was looking at. I said, it doesn't really look like what I know from the Bay Area. I read some papers by biologist David Wake, who has passed away, but he was like the god of lungless salamanders working at Cal. And I read some papers by him that said, we don't search that region, there aren't any terrestrial salamanders. So I said, aha, you'll probably want to know about that. Anyway, that sort of helped segue me from a casual hobbyist herper and scientific illustrator into kind of doing this semi-professionally. On to my work at the zoo. So at SF Zoo, we are raising 3 species right now. We're a Head Start program. We're not breeding any of these animals. We receive animals as eggs or as tadpoles. It's much more beneficial to receive them as eggs because then we don't have to treat them for chytrid. If we receive tadpoles, when they morph out into frogs, we have to treat them for chytrid because pretty much all bodies of water at this point in California have it, and any amphibian you're picking up probably is carrying it. So we've got Sierra Nevada yellow-legged frogs, which have been split from the southern mountain yellow-legged frogs. They're present in the Sierra Nevada. Well, we can just go to this next one. Present in the Sierra Nevada, sort of north of Sequoia Kings National Park, and then the southern species is found all the way down into LA. These are historic range maps, so a lot of these ranges are larger than what their current distributions are, especially for, say, the red-legged frog. Most of their populations have been extirpated from Southern California. They're very spotty. And then the Yosemite toads, which are along the Sierra Crest, high elevation lakes. And just out of college, I had a grant to live in the park, and I worked alongside survey crews who were doing work with yellow-legged frogs and Yosemite toads. So the conservation status of these animals, yellow-legged frogs are endangered, all the rest are threatened. We also at the zoo raise fork-tailed damselflies, which are native to several seeps in San Francisco. I don't know who's doing that work at the zoo. I've never met the person who's doing it. I've never, in my 2 years there, never seen one. So I believe it's still ongoing, but we are not responsible for that. We are the frog farm. So up to this point, we've released about 3,500 Sierra Nevada yellow-legged frogs. We currently have about 2,000 that we're raising, and they'll be released this year. We have populations from the northern area of the range around Plumas, where they can actually be found in streams and not just high altitude lakes, all the way down to Sequoia Kings, which is about the border between where they split off into the southern species. Up till now, and the program's a little over 10 years old for frogs at the zoo. I've been there too. We've raised just over 2,000 red-legged frogs and released them. They get released in Yosemite National Park. They were extirpated, meaning wiped out of Yosemite Valley sometime after the gold rush. Introductions of invasive bullfrogs didn't help that at all. So if you're looking for summer jobs in herpetology, you can actually sleep in a bunkhouse all summer and have the night shift going out and hunting bullfrogs. So that we, yeah, sometimes I think about it just so that I can like live in the mountains again and dip my feet in the rivers. It's not a lucrative position, but if you want to live in the mountains, it's basically like a paid internship. So we've released about 2,000 in the park, and from what we can tell, they have been spreading out across the valley. There are several higher elevation points that we release them at as well, not just in the valley. Interestingly, we've got about 2,500 in the facility now. We just received some egg masses, and we were told that they were going to be 150 tadpoles we needed to release for this one group, which is actually from Solano County. And they all hatched out, and we just separated and counted them, and it turned out we had 762, which was maybe a little much. My boss wants them to be released, and I told her to renegotiate the contract. Pay us. And then Yosemite toads, we have not released any Yosemite toads yet. We're the first sort of organization that's ever really raised them in captivity, so we're still building husbandry protocols for them. But we've got 2 cohorts from several populations equaling about 250 animals. So we've got toadlets that morphed out in the fall, and then we have 1-year-olds. And the toads actually grow much more slowly. Our facility at the zoo is sort of separated from the main zoo. You wouldn't see us on display anywhere. We took over the old Avian Conservation Center, which was built for bald eagle rehab work. And once the eagles were delisted, they were trying to find something to fill their conservation budget with. And we've done western pond turtle work as well, but working with some folks who had frogs at SF State, Sierra Nevada yellow-egg frogs, and needed a place to put them after they had finished with their chytrid studies. We sort of adopted them, and we had a closet-sized space with a few fish tanks in it with less than 20 frogs. And these are old aviaries that have been converted. They put roofs on them, and that's that building in the back. And it's still an aviary. In two of the wings, we still share the facility with the bird quarantine area. They say it's to keep the birds healthy, but they only put the loudest ones up there, so I think they're just annoying. So it's like kookaburras and hyacinth macaws and eclectus parrots, which are just crazy loud. And then peacocks. So sometimes when there's bird flu going through the area, everything has to be quarantined. So they put the peacocks in the building. So you'll be in there cleaning a fish tank and you'll hear peacocks from the room next door. Some of our facilities, tadpoles are all raised in these large fiberglass tanks. And after the frogs morph out, we either house them in large terrarium aquariums, which were kind of originally what they decided was the best way to raise them at the zoo. We still have them, so we still use them, but we actually find these large plastic stock tanks work better for our purposes because we have a pretty high density of animals. The idea is to raise them as quickly, as cheaply as possible, and to get as many out as possible. So it's, high density. We're literally farming frogs. And then down here you can see all these bins, which for years when I was herping and just keeping critters, salamanders and stuff, I had piles of these plastic bins, and I just thought I was doing my best, but it turns out it's the industry standard. Gasketed Tupperware bins. But when we're doing treatments for our frogs and yellow-leg frogs, also we tend to put them through an inoculation period. So we give them on purpose. We give them chytrid and then heal them, medicate them until they're cleared of the infection. And that is so that they aren't naive to the infection when we put them out in the wild, and it does seem to boost survivability. So red-leg frog.
State frog.
This is our state frog, and it's the jumping frog of Calaveras County from Mark Twain's short story.
Oh, really?
Yeah.
Cool.
The interesting thing about red-leg frogs is they do carry chytrid, but they seem to be able to clear it by themselves, so we don't even have to treat them for it. I don't know what it is about their physiology that allows them to do that, but they carry it, and it's never at loads high enough to cause mortality, which is helpful. So as I said, we get them as egg clusters. They actually hatch out fairly quickly from egg clusters, and then we separate them, count them, and then move them into these larger stock tanks, where we then keep them till the summer when they morph out. And every once in a while we'll have stragglers that just want to stay as tadpoles forever and ever. It's kind of obnoxious. So here's after the egg mass is all hatched out. And the little green lumps in there are tadpole food. It's a Rapashi aquarium product called Soylent Green. But it's just a gel medium like you'd feed crickets. And we needed more tops for our tanks. So on the left you can see the black tops are the old tops, and they have their issues. There's a supporting structure underneath the middle where I'm like, why would we do that? That's where we want the lights. So we have to hang the lights above it, so then the light is all on the top and not in the tank where you want it. You can see how well lit it is in there. Anyway, I was very proud of that, so I had to. Instead of heavy 2x4, we used aluminum tubing. It's just far superior. But you can see the density of animals. That looks like, those are about 2-year-old frogs. The red leg frogs we keep for several years. We try to raise them up big enough so that they aren't just gonna get chomped by garter snakes right away, because that does seem to be an issue in some of the higher valleys in Yosemite, where the garter snakes have been eating bullfrogs for so long that they've gotten pretty robust. So we try and not just feed the snakes. So the red leg frogs stay for several years. And they're very variable in color. On the right there, that's kind of the screaming bright red, and then a more brassy, bronzy one on the left. But we do get some interesting aberrations, which you probably never see in the wild, but because we have literally thousands of them, and we're raising them from tadpoles, they don't get weeded out. So we have some sort of weird stuff happening. Like, this guy's xanthic. I wouldn't be able to tell you what's going on with the other ones. One on the upper left looks like it's lacking any red pigmentation. These two frogs are actually the same frog, going light to dark. This is when you just pulled it right out of the tank, and that's after putting it in a bright white bin for a little bit to get it to show its color. But you can see on these three, there's no pigmentation in the eye, so there's something going on there. We also get albinos, but there's something developmental with that gene that's causing albinism, where they will grow all 4 feet and look very froggy, but they never absorb their tails, and they never grow lungs. They still have gills, so they just stay as sort of near metas forever, and eventually just sort of peter out because they're not eating frog food and they can't eat tadpole food anymore. We're not really quite sure what's going on, but that's something for the folks at SF State to figure out, some grad student. Here are red leg frogs. These guys are about to be released. They are fairly large.
How big do they get?
Red leg frogs get pretty large. Not as big as bullfrogs, but you know, body size, about the size of your hand plus legs. I've seen some very large ones actually out here on Patterson and Corral Hollow.
Largest ones I've seen were out there.
Yeah,
By far.
yeah.
Do you think the size has anything to do with the chytrid resistance?
I don't know. I think the chytrid resistance with them has more to do with the fact that they live in like soup made by cows. You know, they're in these like cattle wallows, so they can handle like. And we're fairly on top of our water quality as far as keeping ammonias and nitrates out of it. But you know, we always discuss they're like the red leg frogs. If you have to skip a water change, the red leg frogs are probably the ones to skip, yeah, based on where they live. It may have more to do with the fact that they can slough their skin very quickly, so they seem to just be able to get rid of it. Is that a question?
Yes.
Yeah, go ahead.
Do you have any pictures of any of the albinos?
I could probably find you one, but I won't interrupt the
Alright,
the
fine.
top. I will show. I had one. It wasn't a great photo, so I took it out. But I will show you afterwards. I'll
Alright.
dig in the files. It basically looks like a little yellow frog. Speaking of little yellow frogs, this is a Sierra Nevada yellow-leg frog. Similarly raised to our red-leg frogs. We keep them in stock tanks. The yellow-leg frogs are far more aquatic than red-leg frogs, so we keep them more aquatically. Instead of having sort of a half land, half water tank, we give them these turtle docks. It seems to work quite well. Cute little guys, high density, like I said. There are reports that they used to be that density in the wild, walking around high altitude lakes, and you had to really be careful not to step on them. And then at a certain point, they just started disappearing, and chytrid seems to be the thing that's really driving extirpation of this species. Yeah, and people are probably familiar with that photo. That's a Jules Sartori photo. And that's from decades ago, but that was sort of kind of the canary in the coal mine for like, what is going on with our frogs? Because you go to these high altitude meadows and there weren't any live frogs and they're all just dead in the water. And basically what chytrid does, it's a fungal pathogen and it infects the skin, and it basically sort of creates like a callus, you could say, on the skin of the animal. And because amphibians breathe through their skin, it interrupts their ability to regulate blood oxygen levels, and they also are absorbing liquids through their skin. So it pretty quickly will kill whole populations of frogs. So we have to treat them. So treatment is little 10-minute baths for each frog in an itrafungal, itraconazole, which is actually veterinary medicine. It's an oral medicine for cats. I don't know about cat fungal pathogens, but it does the trick. We just give them a little 10-minute bath. A question?
So, in the end game, there's no way to eradicate that from nature, right?
Nope. No, there isn't. So, and in Central America, South America, it's been pretty devastating.
Okay.
In like Central America, sort of higher elevation cloud forests that are cooler, it's just wiped out so many species of frog. It's crazy. And you know, researchers talk about like, I was studying this frog for college, and I went back 2 years later to finish my PhD on them, and I couldn't find my study animals. And it pretty much it's chytrid. And there's actually a salamander chytrid that has sort of reared its head in Europe and wiped out populations of fire salamanders in the Netherlands. That's actually how I got started doing husbandry conservation, because what I care about is salamanders. The frogs are great, but I knew this program already existed, and in about 2017, I started with a team looking into the feasibility of starting a captive husbandry program at the Academy of Sciences for salamanders, just in case, because it's not really a question of if those pathogens get here, but when. And you know, the best time to learn how to take care of these animals is before that bomb goes off. We actually got fairly far. I was surprised we got grant money to even look at it. But they gave us grant money to look at it, and then there was a big shake up in the management at the academy, and all new projects were binned, and everyone kind of was like, well, we got other jobs to do. We're all adults who need to make a living. So that sort of petered out. And I don't know that they're doing anything with native animals at all at Academy of Sciences, which is a shame, because they've got a great facility, and they're a fantastic institution. I know they have some exotic newts, like Iranian newts, I think they're raising, but I'm not sure if they're raising them for release, although they seem to be fairly successful. So yeah, and it's easy when you've got 4 or 5 little frogs that need to be treated. For 10 minutes at a time, you just put each one in its little tub with its medication and let it sit. But very often we're doing whole cohorts at a time, so that's like 200 frogs. So when we're doing inoculations like this, we basically have one staff member in a room all day doing water changes and treating these frogs. So in the springtime when we do that, it's all hands on deck. Oh yeah. And just a little video to see, he's not freaking out. It's probably not very pleasant for an animal who breathes through its skin to be sitting in antifungal bath, but you know, he's not exhibiting escape behavior. So, and it really does do the trick. When you find animals that seem to have sloughing skin, their eyes sort of clouded over, they're not doing well, and after one bath, they've sloughed their skin off and they're bright again. We usually do a bath a day for 10 days to be safe. Usually we consider them cleared of the infection after 5, but we do 10 just so we're not having to repeat the process in a month because we accidentally gave all our frogs chytrid again. One of my favorite animals, and definitely my favorite animals that we work with at the zoo are the Yosemite toads. This is Antoadney Hopkins. He's sort of our ambassador. He was confiscated by Fish and Wildlife from someone who had him, was living in the Santa Cruz Mountains when the CZU fires went through. And he, I guess, just gave this up to, it was like, hey, I have this toad, I'm not supposed to have it. And we ended up with it because we're the only institution that keeps Yosemite toads. And they're sexually dimorphic, meaning the males and females look very, very different. The males are sort of this yellow with little spots on them. Actually, Antoadney is quite a spectacular looking male toad. Usually they kind of look like little brown turds. And he's actually got beautiful patterns. So we're glad we have him. He lives in a tank. He's cohabiting a tank with a red-legged frog who, Tiny Tim, who is another ambassador animal we have that can't be released because he had infections in both eyes and had both eyes surgically removed. He's fine. We feed him by hand. He eats, and frog and toad are friends, and they live in this tank together. So when we have folks come to, like I said, he's our ambassador. But they start off super tiny. That's actually pretty big. And that's about the size you'd release them at. And that's not quite full size. These little toadlets. This is definitely the cutest life stage for the toads. My finger comes in here in a second. It's to show you some scale. It's kind of alarming, actually.
Yeah, they're small.
Wow. Yeah, one more just because they're so adorable. Yeah, and they're these little black things forever and ever. You see, I can't even. Don't squish them. And they don't start displaying that dimorphism until roughly a year. Until then, they look very much like little baby western toads that you would find out here. It's almost impossible to tell the difference between the two unless you're in like a known population of them. Just little toad piles. This is how they'll look when you come in the morning. This guy over on the left knows something's up. He's ready to be fed. Great camera work, Ben. And now you can see sort of the males are the yellow ones, and the females are the spotted ones. The lights we have, the heat lamps and stuff we have over the tanks, give everything a terrible green color cast, so I apologize for that. They all look kind of mustard colored in that photo. But they start off super, super tiny. Here's Antoadney again. He's a good guy. Yeah, he's a good guy. Eat your mealworms. So, yeah, when we're feeding tadpoles, what we found is we're trying to simulate algae growing on things. So the Soylent Green, which is a sort of a gel medium, you mix it up in hot water like gelatin and then cut it in cubes and feed it to them. We also use this Ceramicron fry food, which is for really tiny fish fry. And the way most people would use it for fish is just to sort of sprinkle it in like fish food into the water. And the way we use it is a little different. We spread it onto slides, or when we have enough, too many tadpoles, we'll put them on Pyrex plates, and they just mow through that stuff. With the red-legged frogs, what we found is even though the literature says that they are herbivorous as tadpoles, they are quite cannibalistic at a certain stage, and when they start to pop feet and legs out, they'll start to chew each other's toes off. So yeah, so it's a trade-off between water quality and feeding them, but we try to give them things like bloodworms, brine shrimp, and stuff like that to sort of keep that bloodlust down. Also, they clearly want that sort of stuff. They're going through big changes, reconstituting their DNA and then becoming frogs. So the protein is probably very helpful. And then when they're adults, all those boxes, each one of those boxes is 1,000 crickets. So typically when we are at capacity, we're feeding out 70,000 crickets a week, and we still run out very often. And it's someone's job, and by someone I mean it's me. I always do it. Someone has to process those crickets and put them into feeder bins so that they can get gut loaded, which takes most of an afternoon every Tuesday. And even though we hire new people and interns, they keep giving them Tuesday off. So like I said, I'm the one who does that. But I actually think that's part of how I got the job is I mentioned that I had a background in culturing invertebrates. So like our toads, when they morph out into toadlets, are so tiny that even pinhead crickets are too big for them. So we found starting them off on springtails and then working up through different sizes of fruit flies, and then occasionally giving them bean beetles, which are also small feeders, until they can get large enough to eat crickets. And I had a background culturing fruit flies and springtails because I often kept very tiny salamanders that needed that. And I could see their ears perk up in the job interview when it said, I raise springtails. So like Edgar said, having those skills in your pocket, who knew they were marketable job skills, but it turned out. Water quality. So we have 2 500-gallon cisterns that produce reverse osmosis water for us. So we take everything out of the water, which is actually bad for animals. So we have to put stuff back in the water. But it's the safest way to make sure that we're taking heavy metals, chlorines, chloramines, and all that stuff that is, it's not great for frogs, but it's absolutely deadly for tadpoles. Like even blue nitrile latex gloves are bad for tadpoles. They leach things, and we've actually even had some problems when doing pit tagging on frogs where batches of nitrile gloves have proved toxic to frogs, and we had mystery ailments that we could only trace back to a bad batch of gloves. So water quality and you know, keeping chemicals out of their enclosures is pretty important. I don't have a better picture of this cistern, but it's, you know, 4 times the size of me. It's about the size of a Volkswagen Beetle. And then all these systems that I'm trying to learn better, we always just call it a technician in, although they don't always make me very confident. I told the last guy who came in to fix a system, hey, I'm not a professional plumber, but I plumb all these tanks, so I kind of know what I'm doing. And he said, I'm also not a professional. And I was like, what? I don't think he meant that. Water quality. On the left, you don't want your water to look like that when you test it. On the right, you want it kind of to look like that. Anyway, the things we test for mainly are ammonia, nitrite, and nitrates. If any of you have kept aquarium fish, you kind of know that cycle. Ammonia builds up in the water first from just waste, animal waste products. Bacteria start to colonize that water and break down those ammonias into nitrates, and then different bacteria break those down into nitrates. And if you have that cycle going well, you don't have to do water changes because the bacteria in the water are actually cycling through the ammonia. And ammonia is definitely the worst for the animals. Nitrates they can sort of deal with. But like I said, you don't want your water looking like that on the left. That's no good. And then before release, we pit tag all our animals. So this is at the veterinary hospital on grounds we get groups of biological consultants coming in who need the training. It's something that they can pad their resumes with, and it helps us out a lot because we have a lot of animals, and it basically takes all day to pit tag animals. I don't have any good footage or photos of it, but it basically involves making a tiny incision on the back of the frog and then giving it a microchip. It's like what you give your cat or dog when they go to the vet, only much, much smaller. It's smaller than a grain of rice. and amphibian skin is pretty resilient. They are healed up fully within 24 hours, and the incisions usually heal within a couple hours. Every once in a while we do find loose pit tags in tanks, or when trying to read the pit tags on animals, we found that they've somehow shed them out. Toads actually eat their shed skin, so a lot of times they'll end up eating the pit tag, and then we're sucking it out of the aquariums with a siphon accidentally. Anyway, but yeah, you can imagine that pit tagging 2,000 yellow-legged frogs, thousands of frogs is pretty labor intensive, and we still need people to do the day-to-day husbandry of water changes and stuff. So challenges for releases. So every animal gets their own deli cup. So we have to sterilize all the deli cups, and then we order sterile gauze pads for each deli cup, and they come in a sheet, so we have to cut them with scissors to fit. We'll find out a better way eventually. Someone bought us like a stamp for like paper craft, but it didn't work at all. We thought we had, and then yeah, we got to collect them all in bins and put them in coolers so that they can be shipped off. And sometimes we're just driving them up to the mountains to meet with our folks at US Fish and Wildlife. But sometimes, yeah, sometimes they get jet fuel, which is very expensive. That helicopter probably costs like over $2,000 to run for the day, maybe more if you count what the pilots actually are paid. Mm-hmm. But there's no good way to get these animals to these high elevation lakes by foot. You'd probably have a lot of losses, or you'd need mule teams to get coolers up there, which is just, at that point, just hire the helicopters. And somewhere I have video of helicopter pilots letting frogs go while the helicopter's in the background. I couldn't find that. I really wanted to from stock because I thought it was hilarious. Because Jesse and Jamie, they're in the middle, are my coworkers, but they don't get to go on the helicopter. So the helicopter pilots and maybe some US Fish and Wildlife guys get to go with them. So, you know, they're burning fuel, so they get up there, and the pilot and co-pilot get out and start releasing frogs out of their deli cups. So, some other species that we occasionally raise and rehabilitate. I was talking to Edgar about this earlier. One year as bycatch in some of our yellow-legged frog tadpoles, we accidentally got Sierran tree frogs. And they were in such a crappy little tub forever. So I got permission and scrounged a nice glass aquarium from some other department and put together this nice little vivarium for them. They're sort of in that room with Antony Hopkins as our ambassadors. I told him to put it in the office with the PhDs because it would be a nice little office pet, but they said no. And San Francisco garter snakes, we're actually working with, gonna just slurp down that worm. We're working with SF Airport. They've got some construction they need to do, and as mitigation, they're hoping to have a breeding program set up at the zoo, so we're gonna probably take that over, although it's probably a year or two out. And as Edgar was talking about earlier, the Germans have them. Years ago at the zoo, they were setting up husbandry protocols for SF garter snakes. And they had a few wild caught ones from the area, but they were worried about getting a genetic bottleneck. So they actually went to Germany and shipped in captive bred SF garter snakes so they could get more expansive genetics in the program. So we actually have protocols for raising garter snakes. We just need to find the space. This guy, they were doing some ditch clearing at SF Airport. So probably heavy equipment, bulldozers and stuff. And you can sort of see right there, a shovel or something nicked him and he had a pretty big gash in him. So he came to the zoo and got sutured up, and then we kept him for several months. We fattened him up on earthworms and pinky mice, and then he got released. And the other thing we keep are western pond turtles. So we've been working with point rays and some areas in Lake County, collecting eggs, hatching them out, and then raising them to an appropriate size to release. Although last year we got 4 eggs, and I think we ended up releasing 2 turtles, so it was not a robust release last year. And it's given the drought that we had been going through over the last decade, the finding eggs is pretty tough. We do occasionally also get rescues. This guy on the right was a turtle that was found in point rays, and you can see he's missing all kinds of parts of his shell. It looks like a bear just picked him up and was chewing on him. You can see the indentations. He was missing an eye, so we named him Odin. And fattened him up and made sure he was healthy, and then he got released as well. The most turtles we've had, when I worked there in the last 2 years, we had a group of turtles from Seminole Regional Wilderness that were all found around a pond that had completely dried up, and when they were x-raying these turtles, they found they were completely emaciated. So we had about 20 of them over the course of a year that we kept and fattened up. But they're actually fairly picky eaters, which is surprising for a turtle, since they just eat dead things. So it took them a while to fatten up. There's also there's a fungal pathogen that's affecting turtles as well, and western pond turtles seem to be getting it, and it affects their shells and rots through their shells all the way down to their body cavities, and it is also very deadly. They're still sort of researching that. It's a new menace. Anyway, that's what I've got. Special thanks to all our partners. U.S. Fish and Wildlife, Oakland Zoo helps us a lot. They're also doing similar work. They're working with southern mountain yellow-legged frogs and foothill yellow-legged frogs. Yeah, and with that, I'll take questions.